First is introduction. All living things found in an environment are considered to be its biotic components. A population is a collection of organisms belonging to the same species that are present in the same habitat. A community is made up of population of several species live and interacting in the same space. A set of every species present in the area and a comparison of population size is one method for researching the interaction between organisms in a community. In this practical, we will use a simple concept of quadrant. A quadrant is a square or rectangular part used in ecological research to study the distributions, abundance, and diversity of organisms within a defined area. Quadrants help in studying specific habitats, identifying patterns of species occurrence, and understanding environmental influence on species distribution. The objectives in this practical are to introduce the simple concept of quadrant, to enhance the power of observation, to record the difference in plant distribution within the quadrant, and to infer from natural observation the possible factors that affect distribution and abundance of organisms. This is the material that we use in this practical, which are raffia strings, measurement tape, stick, scissors, and notebook. And these are the procedure of this practical, and the next video will show the activity of what we have done in this practical. This is the results for this practical.
Next, for discussion part, the difference in the number of plant species in the quadrant. It is because the influencing factors that could have caused this type of distribution. Several factors may have an impact on the variation in the number of plant species in the quadrant, resulting in an uneven distribution. To explain this kind of distribution, a number of theories or presumptions might be made. Firstly, environmental heterogeneity, in which changes in soil properties, moisture content, solar exposure, or microhabitats within the quadrant may allow for emergence of distinct plant species. Certain conditions may favor some species more than others, resulting in their dominance or absence in particular location. The factor may include competitive exchanges. Plant species distribution may be impacted by competition for resources like nutrients, water, or light. Lower species richness in some section of the quadrant may be caused by dominant or aggressive species that outcompete or hinder the growth of other species. Some plant species may have limited dispersal capabilities, which might affect distribution within the quadrant. Variation in species composition can be influenced by factors such as seed availability, dispersal method, and dispersal obstacles. The next issue to consider is biotic interactions, in which interaction with other organisms, such as herbivory or mutualisms, can impact the distribution of plant species within the quadrant. Herbivores, for example, may preferentially devour certain plant species, resulting in their depletion. Finally, there may be a random chance component in the distribution of plant species inside the quadrant. The interaction of environmental elements and ecological process can be complicated, resulting in a stochastic pattern of species distribution.
In conclusion, this practical was intended to improve our observation and recording skills while also introducing the straightforward idea of quadrant sampling. We were able to methodically monitor and document the distribution and abundance of species in their natural habitats by using quadrants. We were able to deduce possible factors affecting these organisms' abundance and distributions based on our observations. we learn a great deal about the ecological processes of the environment and the investigation by using this approach. These results highlight the value of systematic observation while also highlighting the significance of comprehending the factors affecting the structure and operation of ecosystems. Without a doubt, more exploration and development of these observational methods will advance our knowledge of ecological processes and encourage effective management and conservation techniques. The mission is to define the term quadrat in practice. Quadrat is an ecological sampling unit, consists of a small square frame that is laid down which all species of interest or specific area of the community to be sampled, and the measurements are taken. Within the quadrat frame, the existence of plants are recorded using an appropriate measure of abundance. For example, it can be used to measure the abundance of various plants within the quadrat frame. What are the two basic requirements of all the quadrats technique? First of all, area of quadrat must be known. It is essential to know the exact area of the quadrat being used in the sample technique. The area of the quadrat is typically determined by measuring its dimensions, such as length and width. Knowing the precise allows for accurate calculations of population density or other ecological measurements within the quadrat. And the second is the population in quadrat must be determined exactly. To calculate the population density using a quadrat, the number of organisms or target samples within the quadrat must be determined precisely. This requires careful counting or identification of all samples belonging to the target species or organisms within the quadrat. What would be the advantages and disadvantages of using a larger or smaller quadrat to survey sedentary organisms? Larger quadrat can give more accurate estimation of overall densities and allow us sampling species populations spread out over large area. However, its disadvantage is the sampling process will become more complicated and need to spend more time to finish sampling, while smaller quadrat is very easy to use and not take more time. But it can be sampled on a small range of species populations. Problems with spatial scale often make it difficult to directly estimate average densities in habitats such as large forested area or in aquatic habitats where one cannot directly see the animals or plants of interest. Explain the issues concerned ecologists in conducting the sampling method. So, problems with spatial scale make it difficult to estimate average densities in large forested areas or aquatic habitats due to inaccessibility, which can be challenging to access, making it difficult to perform on-site sampling. Factor is heterogeneity, which is frequently strong in these ecosystems and characterized by variance in the vegetation, microhabitats, or water quality. The distribution and abundance of organisms may be affected by this heterogeneity, which makes it difficult to collect representative samples. Next is a scale mismatch. The scale of the sampling procedure may not match the scale at which the organisms are spread or interact. For instance, if an organism displays fine-scale patterns or interactions but the sampling method uses a wider scale, The samples may not be accurately captured.